All right, now that we know everything that there is to know about the history of microscopes, we're going to take a look at how to use our microscopes in class. The first thing you should notice is that there is a cover on this microscope. This is going to keep it safe and clean from any dust and particles that could land on it. The dust could actually potentially damage the microscope, so we want to protect it as much as possible. But for now, we can remove the cover, fold it up, and put it aside so that it is out of our way while we are working. Before we plug it in, I want to draw your attention to some of the aspects of the microscope. The first being the structure. So this area right here, this is called the arm of the microscope, and this area here is the base. If for any reason you actually need to carry the microscope, what you need to do is have one hand on the arm and the other hand underneath the base. This is going to be the safest way to carry the microscope without risking dropping it or damaging it in any way. So now that we've done that, we can plug our microscope in. And when you plug your microscope in, you're going to want to make sure that the cord is not in your way for when you're working. So we're plugged in, we're not turned on. The power button, the power switch, is actually closest to you. So when you flip the switch, you should notice immediately that a light has come on. This is a light microscope, so it only stands to reason that there is a light source attached to it. Now we are ready to look at a specimen underneath the microscope. The first thing we need to do is lower the stage. It's very hard to find the specimen on the slide when you're already very close to the lens, so we're going to lower the stage as much as possible. The way to do that is using this coarse adjustment knob. This rotates, and you are going to rotate it until the stage no longer goes down. That is as far down as it goes. Additionally, you're going to want to put the nose piece, this is a rotating or revolving nose piece, you're going to want to set it to the smallest magnification objective lens. So here we have 3 objective lenses, and on them you can see this one says 4x, so this magnifies 4 times. This yellow one, and I don't know if you can see, this says 10x, so this is a 10 times magnification. And the blue one is 40x, 40 times magnification. So the smallest magnification is going to be the red, the 4x, and we are going to adjust it so that it is pointing down, so that now we have a straight line from the red objective lens to the light. And in between there, we're going to have our specimen, so we can actually look at the specimen. What I'm going to use is a prepared slide. These are non-living cells. We will use these in class, but we will also prepare our own slides at some point in class as well. You're going to put the slide on the stage, and then to secure the slide, you're going to put it underneath these stage clips. Okay, this is just to ensure that the slide does not move when you don't want it to. And then you need to adjust the slide so that the specimen is right over the light. And when you're using the microscope, you'll see that there's a little hole there, a little glass, so that the light can come through. Now we need to start focusing the objective lenses and view our specimen. And what I'm going to do is I'm going to look through the eyepiece right here. Now remember, this is a compound microscope, so there must be more than one lens. The first lens is right here in the eyepiece, and if you can see, it says 10x. The second lens is whichever lens we're using down here. So when you have 10x here and 4x on this red one, your total is actually not going to be what some people would think. Some people would think it's 14 and you just add them. But that's not right. What you do is you multiply 10 times 4. So your total magnification would be 40 times when looking at it under this objective lens. So the first thing I'm going to do is I'm going to raise the stage until I can see the specimen. It's not going to be very very detailed just yet because it's only 40 times magnification, but we have to get it into view first. Okay, so right now it is in view, and all you can really see is some pink because the specimen has probably been dyed so that you can see it better. But we can't see a lot of detail. We want to get closer. We want to look at more detail. So what we do now is we rotate our revolving nose piece so that we have the next highest objective lens. And after 4 times, we have 10 times objective. So now we're going to look at through the yellow, and notice there's a snap to make sure that the objective lens is in place. It clicks. Now we can make sure that we are adjusted clearly. I can see very well right now that this is some kind of tissue, human tissue probably, but I still can't see very much detail. So I want to get closer. And now we are going to adjust the nose piece so that we're on the highest magnification. Now this objective lens is 40 times magnification. So when you're calculating the total magnification in this situation, you have 40 times and 10 times. So your total magnification in this case is 400. At this point, when you are adjusting and you're trying to clear up the image, you're going to use this knob. Don't use this knob anymore. This is the coarse adjustment knob, and this is the fine adjustment knob. You don't want the stage to move really very much at all, because you can already see the specimen. You just want it to be a little less blurry, a little bit more clear. So you're going to adjust it until you can see very clearly exactly what you're looking at. In class, you will actually visualize different types of cells using this microscope in particular. Some of the cells might even be living cells, who knows, whatever we get to. Now once we are finished using the microscope, we need to do all the same things, and it should be put back into the manner that it was found. So the first thing you want to do is remove the slide. You're going to lower the stage, take the slide off, okay, make sure not to break it. If this was a prepared slide that we made, you would need to clean the slide, but you can't clean this off, it's glued down like that. I'm going to turn off the microscope, unplug it, make sure that the cord is wrapped and tucked neatly in here. And then always remember to put your cover back on. Remember, this protects the microscope from any dust and particles that may land on it. And now you are done. Hopefully you remember this when we are using our microscope soon.